process we're looking at here is called transcription. Now, transcription is kind of part 1 out of 2 of something called protein synthesis. And protein synthesis is what occurs with the use of the recipes located on our chromosomes in order to make things inside of our cells, cells of all living things, which give us characteristics and traits, and also have particular functions, make stuff happen inside of us. So if I look at this diagram here, we've got some linear chromosomes. So because they're linear, we know they belong to a eukaryote. So it could be a plant, could be an animal. We would have circular chromosomes if we're looking at bacteria, which is a prokaryote. And we've got particular sections that are shaded in. So we know these chromosomes are made up of long chains of DNA. Now these long chains of DNA have unique sequences because of the bases, the order of the bases, the sequence of the bases that make them up. And these sections of the DNA sequence on the chromosomes are divided up into genes. And we can see here in the diagram that we've got some particular chromosomes that are segmented up, shaded in, which is to highlight how there are different genes on the particular chromosome. So what actually do we need to do inside of the living thing in order to access that gene and turn it into something that will look a certain way on the body of an organism, or act, behave a certain way, and do something inside of the cells of that organism? So what we need to occur is we need protein synthesis to occur, and I'm going to talk in this little video about transcription, which is the first part of protein synthesis. So if we scroll down here, we are looking at a DNA double helix. So we're looking at a particular gene here, and no, keep like we're looking at a particular gene here which has been unzipped or unwound. So what's going to happen is this will occur because of an enzyme called RNA polymerase. This is going to be my rough enzyme. So RNA polymerase is an enzyme. Remember, the enzymes are proteins, usually, that do things inside of our cells. They have a particular role or function. So RNA polymerase is going to unzip our DNA double helix, and then it's also going to help arrange some nucleotides in a little bit. So the first thing that happens is RNA polymerase unzips the double helix, so we have access to the template strand of the DNA. So remember, DNA has a template strand and a complementary strand. The template strand is where our information is, where our recipe for making something happen is. So the RNA polymerase unzips the DNA and gives us access to this template strand. Now what's going to happen also with the help of the RNA polymerase is RNA polymerase is going to go along and it is going to help attach a particular nucleotide to the appropriate base which is being exposed on the DNA. So remembering base pairing rules, we would see something a little bit like this. Now I've left the A for a particular reason there blank, because what we will see is usually adenine and thymine are what cause attractions together, pair together, the bases that pair together in DNA, but we won't see that here on an RNA molecule. An RNA molecule, A adenine actually pairs with U uracil. So the thing that we're making here, RNA, is very similar to DNA, but RNA is able to travel outside of the nucleus and out in the cytoplasm where part 2 of protein synthesis occurs. So this is why the DNA can't just travel out to the cytoplasm and be involved in the second step of protein synthesis. It's not allowed to leave the nucleus, so RNA is going to take it from the nucleus out the cytoplasm where part 2 needs to occur. Now we've talked about how in DNA we have adenine, guanine, cytosine and thymine. In RNA molecules we also see adenine, we also see guanine, we also see cytosine, but we have a U instead of a T. So the U stands for uracil. So there's a different base in place of the T. Anywhere in RNA you see a T, or would need a T, it's going to be substituted with a U called uracil. Now another point just about DNA. DNA stands for deoxyribonucleic acid, whereas RNA stands for ribonucleic acid. Nucleic acid. So there's a difference between the deoxyribose sugar and the ribose sugar in RNA, hence why they get a different name. All right, I know there's a lot to take in. We'll keep going. We'll push on. So once the RNA polymerase has lined up RNA nucleotides, so these are nucleotides, but they're RNA nucleotides, then it's going to join them together, and what we've now seen created is this strand of mRNA. So not only do we have RNA being different to DNA, but we have different types of RNA. So mRNA, the M stands for messenger. So what we see occur is messenger RNA is made off of the base combination on a particular gene in order for that mRNA then to travel out of the, oops, that's a bit shorty arrow, to travel out of the nucleus through nuclear pores and into the cytoplasm, where part 2 of this protein synthesis process can occur. Okay, so just to make things a little bit more technical and confusing even, if we look at an mRNA molecule that we made off the DNA, we don't actually have mRNA as such yet. So we start off with something called pre-mRNA. So I've drawn another diagram here to kind of represent pre-mRNA. So this is our pre-mRNA molecule here. So it's labeled, or called pre-mRNA because it's kind of almost ready, but it's not quite. So on DNA, on strands of DNA, on the template strands of DNA, we have some information in a gene which codes for something being created, and some information which doesn't. So if we look at our particular gene here, we might have a little section here which codes for something being made, and a section here, whereas we might have a segment in the middle which doesn't actually code for anything. It's just kind of nonsensical, and we need to get rid of it. So in order to do so, we need to cut it up. The parts that we need, the parts that we want, are called exons. So I've used blue for exons here. So exons are responsible for actually the coding for the creation of stuff, creation of proteins more specifically, and the red parts called introns we don't actually want. So we don't need the introns. So to turn our pre-mRNA into mRNA, which is what we want at the end of, by the end of transcription, we need RNA splicing to occur. So RNA splicing just involves the removal of the introns to the point where, and we would sort of chop it up like that still, to the point where we just have exons left over. And once this has occurred, we now have an mRNA molecule, messenger RNA molecule, which has travelled out to the cytoplasm of the cell, so protein synthesis part 2 can occur. We've got this information from the DNA, from a particular gene on the DNA, of how to create something, how to create a protein, as the DNA is unable to travel out of the nucleus. All right, so that is transcription. That's part 1 of protein synthesis.